Great, I'd like to welcome everyone. Thank you, Dr. Oskoui, for coming to offer your time and expertise and putting your presentation together for us all to understand more about how evidence-based medicine, how it all translates to application to the patient. I just want to say Dr. Oskoui is so respected, not just within the province of Quebec, but nationwide. She's one of our leaders for the SMA community, and I thank you for that and for advocating for your patients. And there's so many things that people don't even know what our unsung hero doctors are doing behind the scenes. And I've seen quite a bit of it, but I've seen some things recently that you've been up to that I wasn't aware of. And thank you for everything you've been doing. So with that, I'd like to say there is an interpretation feature at the bottom. If you haven't found it yet, you can just click on it and choose your language of choice, and you will be able to hear through our translators if you are speaking in a different language. And with that, I'd like to hand it over to Dr. Oskoui.
Thank you so much for that, Susie. I did my pediatric neurology residency here in Montreal at McGill, and then went away for my fellowship in pediatric neuromuscular disease at Columbia in New York. And it just happened to be funded by the SMA Foundation there. And so I spent 2.5 years kind of immersed in a natural history observational study. So the PNCR that's been used as the natural history comparison now to multiple trials. And I have to say that it was during that period of time, it was from 2004 to 2007, that I was essentially transformed into an SMA advocate. So I think once you meet, it just takes you meeting just one family affected with SMA to then become me, I think, right? I just don't understand any other way. So I am ambitiously going to try to talk about a topic that is dear to my heart, which is evidence-based medicine. So I'm a physician, but I'm also an epidemiologist. And I work as a methodologist for the American Academy of Neurology's guideline subcommittee. So this is the expert group that puts together practice guidelines in neurology. And so it's something that I do on a daily basis. But I thought it would be very helpful for all of you to get a sense of what goes into evidence-based medicine, because we need this to interpret the information around us, right? So a lot of times a trial is, it ends and they say it's positive, and then we conclude, well, how does this help me, right? And so you need to use evidence-based medicine as well. So these are my disclosures. I'm an FRQS clinician scientist. I am paid by the American Academy of Neurology for my work with them as a methodologist. In the past, I have chaired the Avexis Data Safety Monitoring Board, but this is now about 3 years ago. And I'm an investigator on several clinical trials in SMA. So the previous session, Gil Willett from RQMO eloquently kind of talked about the drug approval process, at least as it applies to the province of Quebec. And we know across Canada, I mean, these are amazing times. We now have 3 drugs that are approved by Health Canada, right? And so the picture here on the right really kind of demonstrates how we should all feel about this, right? Which it is, but in practice, this is a lot more how I feel about it. Because a lot of times things are approved, but then it's approved in this kind of sliver of patients, but not really, we don't really understand how it could be beneficial to others or able to access it for all the others. So I won't go into the drug approval process, which has already been covered by Gail's talk. And we'll go over a little bit more the process of evaluating these studies to put them in context into how it actually can help you. So when you look at a study, when you say, okay, the Sunfish trial just ended or the Cherished trial just ended, what are they trying to accomplish in these trials? It's essentially to look at a causal effect between a treatment and an outcome. So if I give drug X, will I get this specific outcome? Will I be able to start walking? Will I be able to come off a ventilator? Whatever outcome that they've used in that study. And so they want to know if in the people who were in the study, do we see that kind of relationship, right? And so evidence-based medicine is really the intersection between the research, the studies, your clinical expertise, right? Your experience of what you know is true in medicine, the art of it, and patient values and preferences, right? All of these things have to come together to then make informed decisions on a daily basis. And I like to use this example because it really drives home the point of why you shouldn't just believe what a specific study tells you, because they often tell you something about a very small portion of people. So if I were to tell you that there's a study published in the British Medical Journal, which is a very well-respected journal, that showed that the use of parachute, it was a randomized controlled trial, and they found that parachute did not result in a reduction of death or major injury compared to jumping off the aircraft with an empty backpack. This was actually published. So do you believe it? Will you now on a daily basis, if you were to jump off, or if somebody you knew was going to jump off an aircraft, would you advocate for them to use the parachute? Or would you say, you know what, there's a study, it was a randomized controlled trial, it was phase 3, stop using it, right? So this goes to speak a little bit about our own internal bias, right? So if you read a study and it kind of agrees with what you previously thought, yeah, I just gave it away there, then you tend to believe what the study said. If it disagrees with what you previously believed, then you're like, well, I don't really believe the study. If you walked into it having zero opinion, which we never do, but sometimes you have zero opinion, you have no idea if something will work or not work, then you'll buy at face value what the study told you, right? And so in this particular trial, if you paid attention to the design of the study, you would see that actually the individuals that were enrolled in the study, the scenario where it was applied, they were not at high risk of mortality because the plane was stationary. It was not at high altitude, right? And so it lacks external validity. So you can take at face value. It's very good to draw conclusions based on if you were in a stationary aircraft, parachutes, you're right, they don't make a difference, right? But it tells you nothing about how parachutes will help you if you are at high altitude, right? And so no one is going to do a phase 3 randomized controlled trial taking people up at high altitude and throwing them off the plane in a blinded randomized fashion, right? And just because that study will not happen doesn't mean that we can't make conclusions, right? We can't make meaningful conclusions. I can tell you about, risks of injury from falls at high altitude, right? Accidental falls, and draw conclusions from there and make inferences to say, well, I would assume that if I fall from, a building of this height and I am sure to die, then if I fall from an aircraft that's high up, it would be the same thing, right? So we can make informed decisions even when we don't have these studies with hundreds of people where we randomize and blind and do all the things that, the drug approval process requires of us. And this is important, especially for rare disease, because we don't have that many people who could participate in those trials, for example. And at this point, now that we have treatments available that are effective, it's not ethical, right, to randomize to a placebo arm anymore. So the level of evidence that we need to generate moving forward to make decisions, it's not going to be that, that perfect study that we're used to. We have to accept something different and still be able to make meaningful conclusions moving forward. So putting this in context, when you look at a study, when you read, okay, so such and such sponsor just finished a study and they found it to be effective, not effective, whatever. What are the elements that you should look at, right? So the population, so who are the people, right? Are they kids? Are they adults? Are they early in their disease, later in their disease, etc., right? So what are the inclusion criteria? What's the intervention, obviously, and what are the outcomes that they looked at, right? So those outcomes actually, especially for SMA, are so different based on where you are in life. So if we look at functional motor outcomes, for kids under 5, you're still developing, right? And so, with or without treatment, your change on those motor, functional motor outcomes is very different and very heterogeneous at an earlier age, and it tends to stabilize later on. Or what's important as a change to you when you're 8 is not the same as what's important to you when you're 25. And so, looking at what are the outcomes, do they matter to me? How long did they follow these patients for? So I know that it'll help me over 12 months, but is that same difference going to be seen over 5 years or 10 years? A lot of these questions remain unanswered. So 2 things we look for. First is, how well does the study reflect the truth within the study? So the people who were in the study, that relationship between the treatment and the outcome, how close to the truth are they about that small reality, right? So just to answer that question, there's a lot of things that we need to look at. First is the comparison group. Why do we make a big fuss about the comparison group? Well, it's because if you really want to know what's the difference of a treatment on an outcome, the best way you could do it, right, is you would take a group of people, put them on a plane at high altitude, have them jump, right? Calculate the outcome. Then you go back in time, you take the same people, put them on the same plane at the same altitude, and now have them jump with a parachute, right? So you have the same kind of counterfactual experience, and then you can really compare the differences between the two. Because there's things that you can measure. You can know the age of the people, their sex, their, outcome on a Hammersmith. But there's a lot of things that we can't measure that maybe are important and could influence that outcome. So you really want to be able to use the same people. So when you use a control group, it's essentially a surrogate or a replacement of this perfect comparison, and you want them to be as similar as possible. But a lot of times what happens is we don't have the best comparison group. So a lot of times when we use a historical comparison group, either we don't have natural history. For example, we never did newborn screening, not treat and follow patients over a period of time, and we will never do that either, right? Because it's not ethical to do that. So, the next best thing is then to look at natural history of affected patients, but it's not quite the same thing, right? And the differences in care. So, how we manage respiratory infections, pulmonary support, nutrition today is different than it was 10 years ago and different from 10 years before that. So when we use natural history comparisons, again, a lot of times it's not quite right. Randomization. Why do we have randomized trials? So controlled means there's a control group, but why do we want to randomize patients? Meaning that, there's a defined probability for each participant to be getting the intervention versus the control arm or the placebo arm or whatever comparison arm is. The reason for that are, like I mentioned, you want to make sure that your 2 groups are as comparable as possible, right? So if you are able to randomize them, then if there's things that you can't measure, it does a better job at equally distributing them between the 2 groups, okay? And sometimes, especially when you have less people in a study, so in rare disease, this is often the case, even when you do things randomly, so you randomly assign people to one group or the other, by chance, there's going to be differences between the two groups, right? Random doesn't mean perfectly equal. And so you have to still look at that. So for example, if you look at some of the early phase 3 randomized control sham procedure trials for nusinersen, we carefully had to look at the table 1 that compared the kids who got the placebo versus the kids who got the treatment to make sure that these important things that can affect your outcome are equally distributed, even if it was done by chance. And if they weren't, well, does it actually create a bias in favor of the treatment group? Or was it in favor of the sham procedure group? And by luck, the differences that were found in those studies actually made the sham procedure a little bit more advantageous to have a good outcome. So it worked out. The bias was against the treatment. So showing the treatment worked, we didn't need to adjust for anything. Allocation concealment. Big word, essentially to say, we want to make sure that, myself, for example, as the site investigator, I have no way of manipulating who gets the treatment or the comparison group. Because if I had the ability, then maybe I would be giving the treatment to the kids I thought were a little bit sicker, who could maybe benefit more. And so that would bias, again, that would just completely make the randomization process void, right? So it keeps things a little bit more fair. You want to make sure as well that you have good follow-up so that it's complete. So if you have 10, 50 people in the study, then, and you want to follow them for a year, then at the end of the year, what happened to all 50 people, right? Whether they stayed on the treatment or they stopped taking the treatment, I still want to know what happened. So for this point, I would say if you do end up participating, or if your child ends up participating in a trial, it's always, of course, your choice to drop out of the study, but please do the follow-up assessments just so that they can at least keep track of how you're doing afterwards, right? Because it'll have a big impact on how well we can make conclusions from that study. Masking or blinding is another concept, right? So you can only do this if there is a comparison group that is concurrent at the same time. So obviously in a lot of the studies where there's no comparison group, this doesn't apply because you know that everybody's getting the treatment, and that comes with its own bias, right? Because you know someone's getting a treatment, you're hopeful that that's helpful. So you can over-evaluate or overestimate certain outcomes. But let's say that it's a concurrent control group, then you want to make sure that those who are evaluating the outcome. So for example, if the outcome is a functional motor scale, the physiotherapist who's been trained to do that outcome, they should not know if you're on placebo or on treatment, right? Because that could bias how they assess you. The only times that that wouldn't apply is if the outcome that we are looking at is so objective that it couldn't be biased. So for example, survival, right? No one would argue that you could be biased in assessing if someone has survived or not. So several examples here of studies that give us good information, but that don't have concurrent control groups, right? Nurture was the nusinersen trial in pre-symptomatic patients, for example. This was the very first study looking at the safety of intravenous gene therapy, for example, right? They had 15 kids, 12 who got the high dose, and there's no concurrent comparison group. But they did use natural history comparison that can then be used to draw some conclusions. So these are the things you look at to see if within the study, those few people that were in the trial, the conclusions that they draw, how truthful is that relationship? Then there's the concept of precision. So precision is about numbers, right? So the first part was accuracy, right? How close are we to the truth? Precision comes to show, do you have enough people in the study that you're pretty sure that your conclusion is what it is, right? So on the left, you see something that has a very precise conclusion, right? But it's really off target. It's not accurate. It's not representing the truth. After that, you see an example where it's not very precise. You're a little bit all over the place, but it is accurate. You're circling around the right place. So here you have a well-designed study, but that didn't have maybe enough people in it. And of course, on the far right is what you're really looking for, something that is accurate and precise so that you can draw conclusions. There's also a big difference that needs to be made between statistical significance and clinical significance. So when a trial ends and the sponsor will put out a press release and say, you know, this was successful, our primary outcome of whatever scale met its outcome, it was statistically significant, and they'll give you like a p-value, which is super significant. Well, you still, as a patient, as a parent, as a physician, need to look at the value of that to make a clinical judgment as to the impact of it, right? Because just because something is statistically significant, if there's a tiny, tiny difference between the two groups and you had recruited enough people that you can for sure see that there's a tiny, tiny difference, well, is that tiny, tiny difference meaningful? And if it's not, then the study is really not meeting its outcome. The concept of external validity is a big one, I think, for SMA. So we talked about studies then taking a sample population, right? You're sampling a group of people with SMA and you're trying to make conclusions about everyone with SMA, right? And so you run into trouble because a lot of times they're very different, right? Everyone is different. There's heterogeneity across ages, across things that are meaningful to each person. And so how do you go from what's in the study to draw meaningful conclusions for yourself, for those you know, for the whole community? You know, the concept of external validity of this person jumping from the plane. I use these two trials just as an example. But I could have put any other trial because almost all of them have completely different inclusion and exclusion criteria. And so, you know, these are two phase 3 randomized with a comparison arm that is concurrent, you know, really well-designed studies, right? But the conclusions, and both of them were positive, but the conclusions that they draw are on different people, right? So the Cherished Trial, which was published now several years ago on later onset patients with SMA treated with nusinersen or a sham procedure, right? They included kids up to the age of 12. And in fact, I think the oldest in that study was 9, I think offhand. And the strength of them or their function was set at a certain way. They excluded kids who were supplemented really through enteral feeds. If they had very significant scoliosis or contractures that could have kind of impeded their ability to improve on the functional scales, they were also excluded. And of course, the outcome that they used was the Hammersmith as primary, versus Sunfish was for risdiplam, you know, similarly designed with a placebo arm. However, the ages of patients were different. So the oldest patient, you know, went up to the age of 25. Their later onset definition included type 3s who had lost ambulation, so were maybe more advanced in their disease. There was no kind of exclusion based on feeding or significant contractures. And the outcome that they measured was a very different outcome than the Hammersmith. So, you know, you have two trials, both found to meet their primary outcomes, but then how do you then go from these to draw conclusions on the general population? This is a table that I had put together about a year ago, and I didn't update it, but I think it has enough information to show you. So the axis is based on age, where the treatment was, the first dose was given, okay? And I divided it by SMA types who were recruited in the different studies. And you see that, for example, for the intravenous gene therapy, and I specify intravenous now, they were all treated under the age of 6 months. The intrathecal study underway under the age of 5, right? Now looking at nusinersen, the placebo-controlled or the sham-controlled phase 3 design studies, you have them both in the infantile onset and later onset SMA, and the age of recruitment across these is actually quite different. So up to the age of 9 really is the oldest who was dosed. And for Sunfish, for risdiplam, the Sunfish Part 2 recruited patients up to the age of 25, right? So for a lot of the other age groups, you see where the data is, right? So it doesn't mean, like I said, for the studies where we wanted to see a sham procedure, randomized trial of, jumping from a plane at high altitude, we don't have the study. Does it mean that we don't know what to do? Well, no, it doesn't mean we don't know what to do, right? So how do you draw conclusions? So, here looking, if you want to look at school-age children and compare it to drugs, well, you have several trials that can inform it, right? So you have the class 1 studies, and you can compare those. But then if you look at other age groups and other information that can support that, well, some have historical controls, some don't have any controls that can help you draw some conclusions. And then, well, you don't just take the study results, right? You put it into context of, well, what's your prior beliefs or how do you interpret it? So if for some relationship between a treatment and an outcome, it's not just one study, it's study after study after study that keeps showing the same relationship, you're a little more convinced, right? Or if you see that there's one study that shows there's a big impact, and then there's another study that shows it doesn't really make a difference, then you're a little less certain of your confidence in what that study is showing, right? The precision, like I mentioned, if there's any kind of biological plausibility, of course, which, for SMN upregulation, there would be. The magnitude of effect, if all of a sudden, kids who in the natural history, none of them were ever able to sit, now the primary outcome is their ability to sit without support, well, that's a huge impact, right? That's a large magnitude of effect. And the direction of bias, like I mentioned, even if you randomize, it's possible that your two groups are just randomly different. And so is it possible that the way that they're different favors the treatment arm, in which case then you would, maybe believe in it a little bit less because you're biased. But hey, if the bias is favoring the comparison group and the treatment still looks better, then you're like, wow, I really should believe in this study. Then there's other factors that we then bring into play to put it in context, right? The importance of those outcomes. So the clinical importance and, are they measuring things that are meaningful? Like Gail was mentioning quality of life. A lot of times this is not measured in these trials, but now more and more it is. The benefits versus harm. So how safe is something to use? How easy is something to use and put into practice? How much variation is there in preferences from one patient to the other on using that drug? And its availability and cost. So hopefully I haven't lost all of you and we're not all feeling like this with all of it. Gaps. So phase 3 randomized with a placebo arm, I think these kinds of studies will become less and less a reality within the SMA community, because most people are already exposed to a certain form of treatment, right? And more it would just be unethical to, have someone not be on treatment or have a placebo arm, right? So real world evidence generation is important. So we have across Canada, the Canadian Neuromuscular Disease Registry that hopefully you've all been familiar with. We have an SMA data set within it that the variables for this have been aligned with other international registries. So we're part of TREAT NMD, which brings together lots of European registries in neuromuscular disease. Now, we're also, as part of a recently funded network called Inform Rare, just a little promotion to have you redirected to it. The goal of Inform Rare is pragmatic trials for rare disease. So using the registries to then conduct rigorous clinical trials and testing out real world outcome measures. So outside of the functional motor assessments that are typically done in industry sponsored trials, where you have to go in that day and see the physiotherapist that day and within a certain time window. And if you're not feeling well that day or you underperform that day, then too bad. That's the result. Real world outcome measures that we're trying to develop are things that look at how you're doing on a daily basis at home and out in the community, right? And also working on defining outcomes that matter and patient-reported outcomes that are more meaningful. And another gap that I really think is a huge gap in Canada that needs to be addressed are transparency in terms of conflict of interest. So what is conflict of interest? So financial or an intellectual relationship that can impact your ability to approach a scientific question with an open mind. And regardless, so a lot of times what I hear is, I'm not conflicted. My primary focus is the SMA community or my patients. And of course I'm rational and I'm not influenced by the lunch I just received, the trip that was just paid for, the 1 hour lecture I just gave and got paid this much money for. It doesn't influence me. I'm a scientific person and I make up my mind. Sure, maybe that's true. Maybe it doesn't influence you, but the simple perception of bias will undermine the public trust in science and the medical community. So I think we have some industry partners on the call, and they are always very frustrated that I won't meet them for lunch. This is the main reason why, is because I want to make sure that my voice is not influenced. They actually track this concept of key opinion leaders. So these are, within the pharmaceutical industry, these are independent experts that they hire, but really are they drug reps in disguise? So these are quotes from an article. Key opinion leaders were salespeople for us, and we would routinely measure the return on our investment by tracking prescriptions before and after their presentations. If that speaker didn't make the impact the company was looking for, you wouldn't invite them back, right? So it's not just, do I pay Dr. X to give a talk? You can give a talk on anything, on whatever you want, and see, does it influence what this person's prescribing? No, it's, let's see how they influence the opinions of many, many in that province or in that country. Although these thought leaders in the profession may not write many prescriptions, they can influence thousands of prescribers, and hence prescriptions through their research, their lectures, their publications, their participation on advisory boards, on committees, on editorial boards, professional societies, and guideline document development. So, if you, in the next few days, listen to a lecture that will talk to you about the results of drug X or drug Y, pay attention to that first slide. of, have they received payment from that industry for whatever reason? And then ask yourself how much you can trust the conclusions that they're then going to give you. In the United States, there's this fabulous website that was essentially a result of a law called the Sunshine Act that is readily available. So you can, go to Google, say open payments, and it'll bring you to this search page. And you can type in the name of any physician practicing in the United States, and it will pop up for the past year or for the past many years that they have data on them, how much research funding they've received from pharmaceutical companies, how much consulting income they've received, if they've accepted a single sandwich at a conference from them, it will all show up there. However, in Canada, we do not have this mechanism at all. CADIF and INES, when they do invite experts, when they do invite patient organizations, there is a disclosure process. And so whether they publish that or not, but they do actually take that into consideration. So, some changes are happening, but if you ask me, not enough and not fast enough. So I will conclude there and open up to questions.
Oh, that was amazing. I have not seen a presentation like this, Dr. Oskoui. I mean, it's just great to share that information. I learned some things here. If you have some questions for Dr. Oskoui, please unmute yourself and go right ahead and ask your question. Janik, I see that you're unmuted, so you're ready to ask a question. Go right ahead.
Yeah, I was just going to say it's tough sometimes to have a presentation that are done in layman's term for us regular people to understand. So you did an amazing job. I just want to say that. Thank you for bringing this to our level, I guess. Not that we're not smart people, but sometimes the medical terminology and everything that goes with clinical trials can be kind of overwhelming when you're starting to swim in this big ocean. So I know we have a lot of kids that are maybe taking and have some treatment on. And I know once the clinical trial, I know they're being very supported and they're very looked at. But sometimes I feel when the drugs is injected into our body, I don't feel sometimes our physicians or neurologists are really well equipped to understand, is the drug actually doing what it's supposed to, like the protein level of SMN2 or the actual impact of the drug on our son or our kids. Is there anything else that would be available to the physicians or neurologists to help them understand or give them access, or even like drawing blood from our kids to further do better tracking or follow-up of our kids? Just a quick questions. I don't know if you have answers for that or not.
Great question. And thank you so much for your compliment, because that I really take that to heart. So the Canadian Neuromuscular Disease Registry, I would say, is probably the database that captures the most people across Canada. And the outcomes that are used there may not be, from what you're saying, the ones that you would want, right? And so through Inform, so I chair the SMA working group for the Canadian Registry, and through Inform Rare. So Inform Rare is funded as a SPORE, so it's a strategy for patient-oriented research through CIHR. And it was an innovative clinical trial design. So the idea is to use patient registries to then continue answering some of these questions into the future. So specifically for SMA, our first step is to now look at the registry and make sure that the outcomes that we're looking at are the relevant ones. And so Inform Rare has patient representatives, has a youth advisory group. We're going to, in the next year, kind of look at the outcomes, right? And there's going to be an iterative process where we are going to reach out through Cure SMA, usually, and Muscular Dystrophy Canada, to reach more patients, parents, to get their feedback on some of these things. So there's ways for you to continue contributing to that. I would say for day-to-day physicians who are treating, the outcomes that are being captured tend to be the ones that the payers, so the provincial, each province requires of them to do. So outside of these, safety labs and the basic functional motor outcome assessment, there's not much more that they have to do. INES for the province of Quebec actually then put out recommendations on, just for nusinersen, what should be the everyday follow-up of patients. And they emphasized quality of life and other things that should be measured. And then there were some initial plans to then mandate to collect this information on a provincial level that then they decided right at the time of pandemic not to continue putting more eggs into. But we are, without funding, trying to still put that information together, because not all of Quebec patients are in the CNDR. So I'm trying to work on being able to do that. Some sites are creating biobanks. So you don't want to be randomly just taking blood samples and CSF samples from people without a specific question. But for example, for nusinersen injections, we do have to withdraw 5 milliliters of CSF before injecting the 5 milliliters of nusinersen. And a lot of times what we need to check for example, the cell count or the protein level for safety, take out 1 or 2 mLs to be able to analyze that. So the rest, instead of throwing it out, we're biobanking, and there's a consent form for that. But most sites don't have that in place. The Neuro has it in place. I know that we have it at our site. I don't know if this answers your question at all, but Jenny,
No,
if you contribute at all to informing this, please, get my email address. Can I put it in the chat, or can someone put my email address in the chat so people who want to participate in this kind of thing can,
because I know here in New Brunswick where we have, it's not a neurologist making the injection, it's a pediatrician. I know once the spinal fluid is removed, it's probably just discarded. So they don't do any tests. They don't draw any bloods. We draw bloods before just to look at the regular testing required by nusinersen, but there's no biobanking or any of that stuff. And we've been asking the questions a few times. When we were doing it in CHIO, I think perhaps they draw some blood on a few occasions, which is being frozen now to maybe do further testing later on. But nothing of that sort here in New Brunswick for sure. So for me, it's always the aspects of the real life data, because you still have adults and other people that don't have access to it. And clinical trials are now done for specific age group and some part of the population. So if you come along and get some real life data, once people are actually on treatment, like a biobank would be great, would help us all, I guess. So, but that's it for me. Thank you very much for your question.
Yeah, we did put your email address into the chat. So if you would like to reach out to Dr. Oskoui, you can do that. I see Jan has his hand up. Go right ahead, Jan.
I don't know if we have a time for last question.
Yes, we do.
Okay, thanks to Dr. Oskoui for that wonderful presentation.
Jan, can I out you right now? So Jan is a co-investigator on the Inform Rare trials.
Yeah, it's a true and a great adventure. I would like to know, Dr. Oskoui, about the database evidence. Do you think it's how much it deserves the rare disease communities, or versus how it could be a tool for government to delay the access of innovative drugs? Do you?
So it's never been part of the discussions in terms of delays. A lot of times, if something's approved with a caveat saying that we then also mandate that the sponsor continues to collect kind of real world evidence. then the registry comes to serve that role. Specifically, we wouldn't want, there to be a Novartis database, a Biogen database, et cetera, right? We want a single database that's a Canadian one that is driven by unconflicted investigators that collect the information the same way for all of them, right? And who have full control of the information that's being collected.
Yeah, and we have a question from Suzanne. Go right ahead, Suzanne.
Sorry, hi. Thank you very much. Again, great presentation. I really enjoyed the slide. I think you said that was your study where we could see per drug, the age, who was involved in the clinical trials. I'm wondering if there's a similar chart available that shows the different primary outcome measures, like to compare Hammersmith, and I forget the other name of the other one, because I do find when the sets come out, they'll say, it changed by this point, but to compare apples to apples, it's hard, right? To make that decision and to understand the difference.
I wonder if that would be something helpful for, I don't mean to add work, but for Cure SMA to have on its website. I know Cure SMA US has a beautiful table summarizing the different compounds and what phase of development that they're in. I often refer to it and then copy it into presentations. But beyond that, so that shows if they're like in phase 1, phase 3, or if they've reached market or if they've been approved. But beyond that,
that was shared yesterday, Dr. Oskoui, the pipeline.
Yeah,
and I have seen graphs, what Suzanne is asking, and I am able to attain that.
That would be great, right? To have that readily available.
Yeah.
Perfect. Thank you, Susie.
Does anybody else have a question for Dr. Oskoui? Just please go right ahead and unmute yourself and feel free to ask.
Yeah, me again. Dr. Oskoui signified how present are the pharmaceutical staff in the process everywhere in the knowledge formation and everything like that. Maybe it's not maybe it could be a dangerous way, but do we have another way? Did you have some solution or idea about how could it be the relation between the industry and the formation, the hospital and the staff?
So the solution is not to, not have a relationship, because I think it's important to have a relationship, right? They are partners. I think without industry, we wouldn't have these drugs, right? Like they are part of the community, right? But I think that it's important to be transparent about the nature of the relationships, right? So that then you can, put that in context. And then I think that there are some leadership roles that should just be completely isolated and not receive direct funding, right? Like I should not receive consulting income or sandwiches or travel expenses from industry, right? Because I chair the SMA Registry. What I say, people say, oh my God, like I believe what she's saying, right? I want to make sure that that is not a biased voice, right? If Kadith or Ines asks for my opinion, I want to make sure that it's my opinion. If I'm going to be on the guideline committee as a methodologist who's going to, let's say in the future, we're going to do a guideline on treatments in SMA, how am I going to, objectively look at the evidence or come up with conclusions if I have these kinds of conflicts of interest? So I think that there's, I'm on the board of directors for the Quebec Neurology Association, so many different ways that because of my leadership position, I can't and will not enter those relationships, right? I've broken all of them several years ago when I started to have these leadership positions. But, you know, I don't think that it means that nobody should have those relationships. They can, right? Especially if they don't have as much influence. But then if they do, they need to just be transparent about those relationships, right? I think it gets tricky when it comes to relationships, not just with physicians, but with patient organizations, right? But from so far, what I've noticed is for most, it's, you know, as long as it is a unrestricted support, and that then individuals can make the decision on, you know, who to invite to share knowledge and information, and be transparent about it. It's been okay, you know.
I think
Thanks
that's a
for
really, really good point about the compliancy rules that, you know, they're strict in Canada for the drug companies. From a patient organization perspective, I feel very strongly, as Dr. Oskoui is saying as well, patient is first, pharma is down the line. We want what they got, and working together with the physicians and with pharma, and, you know, hopefully everybody's got the same goal in mind, and that is treatment to the patients and improving life expectancy and quality of life. Anybody else have any questions for Dr. Oskoui? We are almost at time here.
Are there any comments from Sylvain Bussière or Svetlana from industry on this call? I don't know if there's any others to add to that.
Others, Drang, yeah.
Hey, Sylvain.
Hello, comment ça va? I think that we do.
We're happy to see each other.
Yes, it's been a long time. Bonjour, Yannick. I think that we do need real life evidence in rare disease, definitively. I think that most of people in the scientific environment do agree that it needs to be unbiased, so the database needs to be managed by academicians, which is fine as well. I think the real challenge that we've seen in Quebec, at least, is the fact that it's hard for Quebec centers to be part of a national initiative. There's lots of barriers, and I hope that those barriers do fall apart. And one thing I'd like to add to Yannick's point, you do have a neurologist, Yannick, in St. John, a neuropediatrician like Dr. Oskoui, but the data, everything that's related to research in Canada, outside of, you know, a few centers, it's hard to get clinicians on board because of the resources that are required that are not, you know, suffice, or we don't have the budgets to run it, unfortunately. But thank you, Dr. Oskoui, for your clear and honest presentation. I don't know if you expected me to say something else, but I'm very comfortable.
No, it's good.
Well, thank you, Dr. Oskoui, for the great presentation. It's always amazing to hear you speak. And
your camera's on now.
And I really appreciate the fact that you addressed the partnership and the need for partnership and how they should work. We really appreciate it. Like at Roche, we think about it a lot, and the arm's length approach is important, but also we do agree that we are partners and we would like to support the community. Okay, so
Yeah,
thank you.
This is the third time I'm speaking to Svetlana this week, so.
Great, thank you.
Trang, did you have something you wanted to say?
No, very much just aligned to what Svetlana just mentioned as well. Really the need to partner, but really understand the need also to be arm's length on a lot of the things. So to ensure that there's not this sense of bias, or that you know, physicians are not conflicting themselves. As you know, it's really important as many of these major decisions come along, such as guideline development or even HTA assessments, that there isn't that level of perception, and it's based on the evidence and not based on any pharmaceutical influence.
Yes, I agree. You know, my experience has been that been very professional and very much, you know, patients' needs are important and the goal here for everybody involved, including the payers, to a large part. Some of them need more convincing that it's important to them than others. But yeah, it's been a group effort all along here, and like Dr. Oskoui said, you know, it's amazing to see 3 exactly what that looks like was very much accurate with the dead end. You know, you got a road to a dead end, but we've got the bulldozer that's going to open that up. Does anybody else have something to say? All right, then. With that, I would like to say a very big thank you to Dr. Oskoui for this presentation. I am very excited to be able to share it since it was recorded. I think this is an important one for everybody to see from all levels, and I'm going to be making that available and put it out to the community. So thank you so much, Dr. Oskoui, for your time.
Thank you so much for inviting me, Susie, and for everything that you're doing for the community.
Thank you. All right, have a great day, everyone. See you at the next meeting.